Hello, everyone, and welcome back to Kognito. I am so excited you're here today because we're going to unlock the secrets of an absolute cellular marvel. We are talking about the lysosome. It was first discovered way back in 1955, and honestly, I'd say it's probably the hardest working, most underappreciated little structure in our entire bodies. So let's just dive right into it. Okay, so to really wrap our heads around complex biology, it always helps to use a visual analogy, right? Imagine your cell is this bustling, highly populated city. You've got factories churning out goods, you have highways transporting materials all over the place, and a central government kind of calling all the shots. But think about it. Any functioning city is going to produce waste. And that is exactly where our lysosome comes in. It acts as the vital waste management and recycling center of the cell city. It's basically the ultimate digestive compartment. It breaks down complex molecules so the whole cell stays clean, efficient, and super healthy. Without this recycling center, the whole city would just completely shut down. Section 1, Anatomy and Arsenal. Let's look at the lysosome structure. Now, from the outside, the lysosome has a really simple, pretty much invariably spherical shape. It just exists as a small sac protected by a single outer membrane. But don't let that simple look fool you, because inside that spherical membrane lies a highly potent arsenal. And the magic number here? It's 40. A single lysosome contains about 40 different kinds of hydrolytic digestion enzymes. Think of these enzymes as incredibly powerful chemical scissors. They're capable of cutting down these complex, large, organic molecules into simple, reusable forms. We collectively refer to this whole arsenal as hydrolytic enzymes, and let me tell you, they are ready to be deployed at a moment's notice. But you might be wondering, how do these powerful enzymes actually work without, destroying everything in the cell? Well, the crucial point is that all of these lysosome enzymes belong to a specific class called acid hydrolases. And just like the name suggests, they absolutely require a highly acidic environment to function properly. Specifically, they need a pH level of exactly 5.0. Now, to maintain this extreme acidity inside the inner matrix, the lysosome's outer membrane has a specialized proton pump. This pump continuously uses cellular energy, or ATP, to actively pull hydrogen ions from the surrounding cellular fluid directly into the lysosome. It's such a brilliant mechanism, really, because it ensures the enzymes only become active inside the safety of this highly acidic little bubble. Let's take a quick look at the deadly categories inside this enzyme arsenal. First up, we have phosphatases, which break down phosphate linkages. Then we have nucleases. These are capable of entirely dismantling the phosphodiester bonds in nucleic acids, like your DNA and RNA. We've also got polysaccharide enzymes for breaking down carbohydrates, proteases for snapping the peptide bonds of proteins, and lipases to dismantle lipid fats right down into basic fatty acids and glycerol. Together, this elite team of enzymes can effectively break down almost any biological macromolecule they encounter. No way anything is surviving that. Moving right along to Section 2, the manufacturing line, let's talk about lysosome formation. So how exactly are these highly destructive compartments safely built without accidentally digesting the cell itself? Well, it's a true cooperative effort between the cellular organelles. Here's how it goes down. Step 1, the raw materials, or enzyme precursors, are manufactured and supplied by the rough endoplasmic reticulum, or the RER. Step 2, these safe, currently inactive precursors are sent over to the Golgi apparatus for processing. You can think of the Golgi body as the packaging plant. It processes these precursors into their final enzyme forms. And finally, step 3. The Golgi body literally pinches off these enzymes via budding, releasing them safely into the cell cytosol as tiny little vesicles. And boom, these newly minted vesicles are our freshly manufactured primary lysosomes. Section 3, the 4 stages of life. Let's look at the types of lysosomes. You see, the lysosome doesn't just stay the same forever. It actually operates in 4 distinct stages. We start with the primary lysosomes. As we just saw, these are the newly formed vesicles straight out of the Golgi, and they are holding those inactive enzymes. But when a primary lysosome fuses with a food vacuole from outside the cell, it transforms into a secondary lysosome, also known as a heterophagosome, and that is when digestion actively begins. Then, once the digestion is totally complete, what you have left is undigested cellular debris. So the structure is now called a tertiary lysosome, or a residual body, basically just waiting to expel the waste. And finally, we have autolysosomes, or autophagosomes. These guys form when a primary lysosome fuses with the cell's own dead or injured internal structures to recycle them. And that brings us to Section 4, Mission Directives. Let's explore lysosome functions. Let's compare its basic feeding and secretion mechanisms. It is really fascinating stuff. On one hand, we have endocytosis. This is intracellular digestion. Basically, the cell takes in food from the outside, forming a vesicle. Then the lysosome fuses with this vesicle, degrades the complex food using its hydrolytic enzymes, and releases the simple nutrients right back into the cell to be used for energy. On the other hand, though, we have exocytosis. This is extracellular digestion. Here, the lysosome actually travels all the way to the edge of the cell, fuses with the cell membrane, and releases its digestive enzymes outside into the extracellular space to digest external substances. Pretty cool, right? But wait, the lysosome isn't just a digestive tract. It's also a massive part of the cell's immune defense system, and this happens through the process of phagocytosis. When dangerous microbes or pathogenic invaders attach to the cell membrane, the cell just engulfs them completely, forming a trap called a phagosome. Then, our brave little lysosome rushes in, fusing with the phagosome to create a phagolysosome. Those powerful acid hydrolases are unleashed, totally digesting the microscopic invaders and converting them into completely harmless cell debris. It's literally microscopic warfare happening inside you, and the lysosome is our frontline soldier. Then there is the incredibly fascinating process of autophagy, which literally translates to self-eating. Sounds a bit gross, but it's absolutely vital. Sometimes the cell's own infrastructure gets injured or simply dies of old age, like, say, an exhausted mitochondria. Now, instead of leaving this dead weight floating around taking up space, an isolation membrane wraps around the dead organelle. The lysosome then fuses with it, consuming and digesting the injured cellular components. This clears out valuable space in the cell city and recycles the raw materials to maintain overall cellular health. It is the absolute ultimate in internal housekeeping. Now, I want to leave you with a bit of a provocative thought to ponder today. What would happen to our bodies if these microscopic waste managers just completely stopped working? Think about it. Imagine if that cell city suddenly lost its entire waste management and recycling department. The garbage would just pile up, dead organelles would completely clutter the cytosol, invading microbes would multiply totally unchecked. The result would be a catastrophic buildup of toxic cellular waste, which actually leads to severe genetic disorders and, ultimately, cellular death. It truly reinforces just how incredibly important these tiny, spherical organelles are to our very survival. And that brings us to the end of our fascinating journey into the lysosome. I want to warmly thank you all for joining me today and learning about this incredible cellular recycler. If you enjoyed this explainer and found it helpful, please make sure to subscribe to the Kognito Key channel. Hit that like button and share this explainer with your friends, your classmates, or honestly anyone who just loves the wonders of biology. Keep learning, keep exploring, and I will see you in the next explainer right here on Kognito Key.